Peace be upon you all, and welcome to this amazing event where we are starting today the first episode of Seeing God in Biology with Dr. Fuz Rana himself. And Dr. Fuz, how are you doing?
Ahmed, I'm doing great. How about you?
I'm just feeling great and so excited to start this today. And we'll be talking about many things that relate to molecular biology, and you're the expert here. And um, you have put forward a very interesting idea that we should have some explanation of what DNA is and its structure so that our audience can really follow us through. So you have it. Let's understand more about DNA from you.
Yeah. And we're just going to do a quick overview of the structure of DNA. It's not meant to be an exhaustive discussion, but just simply to give those viewers who are novices in biochemistry and molecular biology just a little bit of a tutorial so they're able to actually really fully appreciate just the marvels of creation that we see, in biochemical systems. And I've got a slideshow here. Okay, so here's a slide showing three different depictions of the structure of DNA. And DNA is actually a very large, complex molecule. And
Mm-hmm.
the way to think about DNA is to think about a ladder, right? So the ladder has these uprights and it has rungs that extend from the uprights. And if you took that ladder and you twisted it in a right-handed manner, you would get get a double helical structure. And that is exactly what we see with DNA in the DNA double helix. But each of the uprights of that ladder actually represent a molecular strand. So if you could envision somebody taking a saw and sawing the ladder in two halves, going right down through the middle of the rungs and separating them, that's essentially what you're looking at with the two strands of DNA, where those strands are made up of subunit molecules that are linked together in a head to tail manner. And that each of those subunits essentially is, again, the building blocks of each of the DNA strands. Those subunits are called nucleotides. And
Mm-hmm.
there are four different nucleotides that are used to build DNA: adenine, guanine, thymine, and cytosine. And so the next slide that we have here shows a cartoon of a typical nucleotide. This happens to be thymine. And you see that it consists of a blue structure that's a sugar, a ribose sugar, meaning it's made up of 5 carbons, and attached to one of the hydroxyl groups in the 5' position is a phosphate residue, and then attached to the 1' position is a nucleobase. In this case, it's thymine, but you could also attach cytosine, guanine, or adenine. And this is what ends up happening to build the strand. Uh, the the nucleotides are linked together through what's called a phosphodiester linkage, where the phosphate on one nucleotide in the 5' position reacts with the hydroxyl group on the next subunit in the DNA strand in the 3' position. So it's a 5', 3' linkage. And you just repeat that along the entire length of the strand, where in this particular diagram, the nucleobases are only represented by letters. Their actual molecular configuration isn't depicted there. And so when we look at DNA, again, hopefully now this particular image that we're going to repeat makes a little bit more sense for those of you that are novices, where you have these 2 polynucleotide strands that, again, align actually in what's called an anti-parallel manner, where one strand goes in what's called the 5', 3' direction, and the other goes in the 3', 5' direction. So they're anti-parallel strands. And then the nucleobases that are in the interior that form the rungs of the ladder from the 2 strands will interact with each other in a characteristic manner, where adenine on one strand always interacts with thymine on the other, and guanine on one strand always interacts with cytosine on the other. Those are called the Watson-Crick base pairing rules. But there you have it. That is the structure of DNA in a nutshell. And hopefully this is helpful information that gives you at least, again, some orientation to the structure of DNA, and will help you better appreciate some of the remarkable things we're going to be talking about in these upcoming episodes.
I guess some common questions for some of the audience will be to understand a little bit more about some of the terminology. So can you in a nutshell explain what does 5' and 3' refer to in terms of this ring that we see in front of us?
Yes, so let me go back and to this particular image. That's a great question, Ahmed. Thank you for asking that clarifying question. What chemists do for molecules that form a ring structure, or even linear molecules, is they number the atoms that are, in this case, that are part of the ring. That way you know what particular atom that you're referring to. And so if you look at thymine, you notice that there's these little 1, 2, 3, 4, 5, and 6 associated with each of the different atoms. And so in thymine, or in the nucleobases, again, there's a numbering system. So for example, you would say that there is a methyl group in the 5 position of thymine. And so that way it's a designation. If you go to the deoxyribose sugar, those numbers are primed numbers, just to distinguish them from the numbers in thymine. So you have, for example, a 1' position, which is where the thymine is bound. If you go to the 3' position, you see a hydroxyl group, or an OH group. And in the 5' position, that's where we see, which is a methyl group that extends above the ring, that's where we see the phosphate group linked. And so when we talk about, in this slide, 5', 3', what we're saying is that the linkage here is the 5', and it's going up to the 3'. And then the next, you see 5', 3', 5', 3'. So that's just telling you essentially the direction. So this particular strand of DNA actually starts in the 3' position, because that's the first functional group that you encounter. So that's really 3' to 5'. If you flip that upside down, you would now be starting in the 5' position, because that's where that's the first position you encounter. And so that's what's meant by 5', 3'. And so there's a polarity to the DNA strands. That polarity, instead of saying top and bottom or north and south, biochemists will use the numbering system. In this case, 5', 3', or 3', 5'.
There's one thing that just drew my attention. If you go back to the ring in the previous slide, so we have the oxygen up there. This is an oxygen, correct?
Yeah, that's right. Yes.
Yeah, and then we have the 1', 2', 3', 4'. In the prime positions, these are carbon atoms, or what?
Yes, those are carbon atoms. So in these molecular structures, each vertex, it would be assumed to be a carbon atom, unless and if it's not a carbon atom, if it's oxygen or nitrogen, if it's what chemists will call heteroatoms, that would be anything other than carbon, then they would actually be written out in the structure. So
like the nitrogen and the oxygen, the nitrogen in the thymine and the oxygen in the deoxyribose, right?
Yeah, that's exactly right. So if you go to thymine, that's a great example. In the 1 position, you have nitrogen. The 2 position is a carbon, but bound to that carbon is a double bonded oxygen. If we go to the 3 position, there's a nitrogen. The 4 position is a carbon with, again, another double bonded oxygen. And then if we go to the 5 position, that is a carbon, and that carbon has a methyl group bound to it. The 6th position is a carbon with a hydrogen bound to it. So
yeah, I get that. How come the 5' position is essentially outside of the deoxyribose ring? It's like vertically on top of the 4 prime. What does that signify?
Yeah, I should have prepared a slide for that, but typically
Okay.
sugars can exist in what are called linear forms or cyclical forms,
Mm-hmm.
and so the numbering corresponds to the linear form, but when the cyclization takes place, one of the OH groups because sugars are basically polyalcohols or polyhydroxy compounds. One of the OH groups reacts with a carbonyl group to form what's called a hemiacetal linkage. And so that's what you're looking at here is a hemiacetal linkage, which is why the oxygen is in the ring. And then what ends up happening is one of the carbons with an OH group is actually now outside of the ring. So it has to do with the way the ring cyclizes in this particular case.
So in the case of DNA, you will always have this phosphate group at the 5 prime, and if we look at the layer beneath or above, then the phosphate group will link to the OH that's protruding from the 3 prime.
Yes, exactly.
And hence they build on top of each other this way. And this
Yeah.
builds one side of the ladder, and the other side is running the other way so that they, like fit together.
Yes, yes. And in fact, if you go to, let's see, if you go to this diagram, I don't know if you can see it very well, but
Yeah.
there's arrows on the double helix, and they're going in opposite directions. And that's communicating that there's an anti-parallel orientation. And in fact, in a few episodes, we'll talk about DNA data storage. And actually, that anti-parallel orientation is critical for the stability of the structure of DNA. So every little detail of the DNA structure actually has a role to play in stabilizing and making the structure of DNA actually robust. So that anti-parallel orientation is really important, and it plays out in very important ways when it comes to things like transcription or DNA replication. That orientation of the strands becomes very, very important.
All right. So DNA doesn't just store data in these nucleobases, but essentially just right out from its very structure, it tells the system that reads it which direction to read it in. Is that correct?
Exactly right. Yes. Yeah. So
Amazing.
yeah.
So it's like, English is left to right, Arabic is right to left. So
Yeah, that's right.
it's determined. If you want to read the language, you know which direction to read it from.
Yes, and in DNA, in the structure of DNA, one strand is English and one strand is Arabic.
All right, and in this case, those colors. So the yellow will always have a green, and if it's a green, it will always have a yellow, and the blue will always have a red, and the red will always have a blue, whatever those would signify out of the 4 nucleobases. So
Yes.
this essentially causes one strand to be like the mirror image of the other, right?
Yes, exactly. The terminology that biochemists use is complementary. The 2 strands have complementary sequences. And this is really very important because if you know the nucleotide sequence of one strand through those base pairing rules, you'll know what the sequence is for the other strand. And this actually makes DNA replication possible because it means that the 2 strands conceptually can separate from each other, and then each of the strands can serve as a template to assemble a new strand, and you wind up with 2 daughter molecules that are identical to the parent molecule, where one of the strands is from the parent and one of the strands is newly synthesized. So interestingly enough, when Watson and Crick published in 1953 in Nature the structure of DNA, they have a sentence in that paper that is an incredibly profound statement that they don't really spend a lot of time elaborating on, but other than to say that the structure of DNA immediately suggests the mechanism for DNA replication. And I'm paraphrasing there.
Wow.
But they when they determined the structure, they immediately had insight as to how DNA replication most likely would occur because of that complementary relationship.
It lends itself to something.
Yes, yes.
And just maybe the final question, since there is one running from 5 to 3 and one running from the other way around, which one would we consider the original and which one is the copy, or which one is the complementary?
That's a great question. I'm not sure that you would think about it in those terms. Mm-hmm. Now, sometimes you'll hear biochemists refer to the sense strand and the non-sense strand. Mm-hmm. And what they mean by that is that when the cell's machinery reads the information in DNA, it will tend to read one of the strands and not the other. And the strand that it's reading is the one where the information for the gene is harbored, and then the other strand is just a complementary sequence. But of course, the sense and non-sense strand is relative. So as you're moving along the length of the DNA, you might have a gene encoded on one strand, and then on the other strand, upstream or downstream, you might have a gene coded. So it's not one strand is totally the sense strand and one strand is totally the non-sense strand. It's distributed between the strands, but that's oftentimes the way, biochemists will designate, locally the strand as being where the information is actually harbored versus just simply being that complementary strand.
So that will be relative to the machine that's reading it, for example. The machine would consider this one the sense and the other one the non-sense, for example.
Yes. And there are certain sequences that precede a gene that will designate that strand, at least in that local environment, as being the sense strand.
Okay, so very, very clear signs of information coding in a very intentional and structured manner, just by looking at how things are structured, just at the foundational basic structure of things.
Yes.
So amazing.
Yeah, and it's digital information, right? Because each of the subunits is discrete, and so it's digital information, and it's all, interestingly enough, there's also analog information in DNA, where analog is continuous information, and so it's even though you depict DNA as a double helix, there actually is curvature in the molecule. Depending upon the sequence, and that curvature actually harbors information as well, believe it or not. So there's a three-dimensional aspect to the DNA.
Is that a new discovery, or because it's pretty new to me?
Yeah, it's a fairly new discovery to recognize that the three-dimensional structure of DNA is actually communicating information, and because it's continuous in space, it's in effect analog information. Right, so both digital and analog information are found in DNA, remarkably.
So I'm pretty happy that we are having a specific episode about how DNA is designed, because now this is getting very exciting. All right.
Well, and this is just scratching the surface. So in a future episode, we'll kind of repeat some of this, but go into even more detail.
Okay, I think this is a great introduction to somebody who doesn't know what DNA is all about. So in summary, it's two strands running side by side, complementary to each other. On each strand, there is like a foundational sugar that is carrying a phosphate that links to a hydroxide, and this causes this strand to build on top of each other. And then from this side pillar stems those little nucleotides that can be one of four digits or one of four letters. Each letter is essentially like a letter in a word, and the other set of letters will mirror it, and then those letters will be like the zeros or ones of computers, but instead of two possibilities, they are actually four. So information density might be heavier here, and this is our molecule, and it even tells the system that reads it which one should be read and which one is the redundant. And you've just told us that it even has some information encoded in the way it's formed in 3D, which is mind-blowing. So great introduction, Fuz. And I hope, so if anybody has more questions about that, just throw the questions at us, and we might as well be answering them in the after show. And let's now see what DNA can also do. There will be an after show after each premiere, so send your questions through the link in the description.